Welcome friends. In a series of video we have been discussing about the various aspects of cell division. We had a previous discussion regarding what is cell division, what exactly happens in cell division, what are the different phases of cell division, and we had a discussion regarding the interphase, what is the basic structure of chromosome, how do you count the number of chromosomes. So we had some very basic discussion that helps us to understand the exact phase of cell division in our previous videos. So in today's video we'll be talking about the nuclear division, that is the karyokinesis, which has got two phases, that is either through mitosis or through meiosis, right? And today we'll be discussing about the mitosis, with which we'll be able to understand meiosis also, okay, because there is no significant difference, okay, in my opinion, between mitosis and meiosis, though there are differences, I do agree, but the general pattern of the nuclear behavior in these processes is going to be one and the same, right? And in today's discussion we'll be talking about mitosis, which has got four phases: prophase, metaphase, anaphase and telophase, right? And we'll be talking about just two phases in short, right, in brief, that is the prophase and metaphase. So this is going to involve various terms, right, for you to be comfortable with this discussion. So kindly get back to those previous video, get used with the concepts and terms, and then join us here, right? Let's begin with our mitosis, which has got four phases, of which we'll be talking about the prophase and metaphase. So we'll restrict ourselves till this particular thing, okay? Prophase is going to be the first phase of mitosis. Here there are two things given: early prophase and late prophase, right? So let's see what exact differences between the early prophase and late prophase. See, first thing that is going to happen here is the chromosome, which are found as threads, as chromatin, okay, which are into loops, that is entangled, it becomes untangled, right? That is, the chromosome becomes loose, they are not twined, right? You get this point. So this is one thing that happens. Second thing is the chromosome condensation begins. Now you could see the difference between the chromosome in early prophase and late prophase compared to the one in early prophase. The late prophase, the chromosome is much more condensed. You could be able to see it as a distinct compact structure. So this is another important event that happens in prophase. And the centriole duplication, you could see two centriole here, centrioles are in doubles. The centriole duplication, which has happened in S phase of interphase, right, move towards the opposite pole, from which the spindle fiber, which is the rays of microtubule, emerges, and you entire structure you call it as a mitotic spindle. So this is given in the transition to metaphase figure, but this migration happens in prophase itself, right? So this mitotic spindle assembly, okay, begins in prophase, okay. And finally you could see here the nuclear membrane disappears and all the other organelles do disappear, right? So these are going to be the 3 to 4 points are going to be the characteristic feature of prophase, right? Fine. And this disintegration of nuclear membrane, just beginning of the disintegration of nuclear membrane, marks the beginning of prophase, right? So after prophase, the cell division gets into the metaphase. You have a separate picture here, that is the transition to metaphase. So what happens in metaphase? You could see when you compare this late prophase and transition to metaphase, you will be able to see two important features, right? One is the nuclear membrane. So here this has got an intact nuclear membrane. Here the nuclear membrane starts disintegrating. Here you don't see the nuclear membrane. So the nuclear membrane disintegration gets over in metaphase. So this is very important point. The nuclear membrane disintegration just begins in prophase, but it gets over in metaphase. This is one point. Second point is the chromosome condensation, which has begun. You could here you see the chromosomes are in, okay, thread-like structure. You could see to an extent the chromosome has got condensed. Here the chromosome condensation is complete. Here the chromosome has two chromatid because they are in dyad, because in synthetic phase the chromosome concentration increases. They have got dyad, okay, which are joined by centromere, which has got two spaces on either end, which is called as kinetochore. Each chromosome has got two chromatid, one centromere and two kinetochore, right? Now what happens? Mitotic spindle, which radiates the spindle fiber, attaches with both the kinetochore of the chromosome. So let me consider this as the upper centromere, this as the lower centromere. So considering this chromosome, so the spindle fiber from the upper centromere attaches here, okay, the other kinetochore is attached with the spindle fiber from the lower centriole, right? So my point is, each chromosome which is in dyad is connected with both the centriole through spindle fiber, right? This is the point, right? From here you shift your focus to the metaphase here, you will be able to see that the chromosome, condensed chromosome which are arranged here and there has got aligned in a straight line, which is what is called as an equatorial plane, right? And each and every chromosome is, as I said, it is attached with the spindle fiber from both the centriole through the kinetochore, and you call this as a metaphase plate. This metaphase plate has got its own significance, right, in terms that it is easy to calculate the number of chromosomes. Say, you always calculate the number of chromosomes by counting the number of centromere. So how many centromere? 1, 2, 3, 4, right? So in case of 4, it will be quite easy for you to even count the number of chromosomes in this phase also, right? But when you consider chromosomes in higher number, when you see that the chromosomes are lined at one after the other, it will be exactly easy for us to locate and count the number of chromosomes. You see a crowd gathered. Once the crowd, they are in a queue, it is easy for us to count the number of people. The same concept is applied here, right? So this is what is called as a metaphase plate, okay, which has got chromosome which are in dyad, that is 2 chromatid which are in dyad connected with the centromere. Each centromere has got 2 kinetochore. One is attached with the spindle fiber from one centriole, okay, the other is attached with the spindle fiber from the opposite centriole, right? So this is going to be the metaphase plate, right? And the chromosomes are also clearly visible in the metaphase. Let's stop our discussion here. In next video, we will begin our discussion from the anaphase and complete till the telophase, right? Thanks for watching.